Hi there, and welcome to the BioHackers podcast. I'm your host, David James Clark IV, here with the man, the myth, the legend, Dr. Alex Feltus. How you doing today, Alex?
Hey, I'm doing much better now. Thank you. I feel good. I feel really good about myself.
You should. You're amazing. And today we've got an incredible topic. So before we get there, cue the space music. All right, that was cool. Hey, you know what? We have Josh to thank for that opening scene. He actually created the title screen for us. Thank you, Josh, and we'll be learning a lot from JVB in just a few minutes. But we're here today to talk originally about the JWST. Something really exciting just happened, and usually we start off the podcast talking about what we're vibing. Today I'm vibing space. Are you vibing space, Alex?
I've been vibing space since 10:30 a.m. Eastern yesterday when they released the images.
Yes. So we're, of course, talking about the James Webb Space Telescope, the JWST. NASA released the most, I guess, in-depth, far-reaching, universal pictures that anybody's ever seen, and they're gorgeous. For those of you watching the YouTube version of the podcast, I'm showing them to you right now. Those of you who are listening, just imagine what space looks like. It kind of looked like a cross between Star Wars and a piece of, I don't know, like pixelated computer art. It's really beautiful.
It's just unbelievable. It's out there and it's endless. It just keeps going on and on and on. And I spent, I don't know, I kind of wasted a couple hours yesterday. I don't know if it was wasted. It was so much fun.
No.
Looking up the Carina, or I'm not sure if I'm pronouncing it, the Carina Nebula. There's a little like birthplace of stars that they showed pictures of from JWST in infrared and high clarity relative to Hubble. And Hubble was awesome, right? That's like a 1990s telescope, but the resolution was phenomenal. And I really geeked out, and I went and found the 1820 star survey where the person had first discovered. I can't remember his name. It was terrible, but he had first mapped it from Australia. It's in the southern skies, and so the resolution, was amazing. How much clearer it's gotten of this one place in the sky, and I wonder what it's going to look like when we go there, and we're going to need food. When we go there, we need people to grow food in space, and that's what today's episode is all about, or partially about.
Yes, that's right. So how do plants interact, grow, and thrive in weird gravity situations, phototropism and gravitropism. I did a little bit of research before the show on, Josh's website and trying to understand a little bit about his lab. He has some plants on the space station, so we're going to talk about that. But yeah, it's just, it's fascinating, the excitement and enthusiasm for this $10 billion telescope, which, is providing, like you said, a level of resolution for these huge nebulas, these, galaxies, beyond our own. And it gets people really jazzed about space. I've been, it's all science fiction, right? So we all imagine through Star Trek and everything, what space is like, but now we're starting to see it. And when you see it for real, did something click inside you? Because I felt this sense of wonder, associated with that I'd not felt before. When it's a TV show, it's exciting and fun, but it's a TV show. Now you look at it and go, wow, that's real.
I know, it's just it's unbelievable. There's one image too. If anybody, by the way, there were like 5 images released, and one of the NASA scientists said that this is a week's worth of work. Like they can do this every week, and what they released was unbelievable. There was one picture that they released where it's pictures of galaxies further back in time, I believe, than anybody's ever seen. I'm not an astronomer, so I don't know exactly what's all true here. but a lot of those galaxies are bent and diffracted, and they're pulled apart because they're going through gravity wells from dark matter, which you can't see. So they're kind of warping around that. And you could see, like, you go out and look in the sky, and we see stars here, but if you look out further in the sky, if your eyes are better, you had a telescope, then you'll actually see everything warped, and that's the way it looks out there. So we're seeing, I don't know, it's kind of like what we're seeing with DNA. Like, we're seeing down to the single base pair of billions of ATGs and Cs in a person, and what's different between different people. Like, we're looking down at that molecular level, and then we're looking out at the universe and seeing the same thing as high resolution views. That, I don't know, it's just we're in the middle of a revolution in technology and science, and it's hard to see sometimes, especially with all the nastiness going on in the world with pandemics and things like that, right? It's like, but there's so much tech and hope out there, and that's what I see. I see brightness for mankind when I see these pictures, and I also see every once in a while, I'll see like Anakin flying around trying to blow up a Death Star or something like that.
Yeah,
right?
right.
I think I got my Star Wars. My Star Wars lore was messed up. I apologize. Oh yeah, that's right. I forgot about that word. Spoiler alert.
Spoiler alert. If you haven't seen the 9 movies, go check them out. So,
exactly.
One thing you've said in the past that I found fascinating is if you look at, it's all about perspective, right? So if you look at some of the distortion fields and some of the formations that these galaxies take, and then you turn that on its ear and you go deep into the DNA. And there was another kind of Marvel film, Ant-Man, where they kind of went into the microverse instead of the universe. And you look at the with high resolution, you look at what's happening at the atomic and maybe even subatomic level. It's very similar looking. I mean, it has similar patterns. Is that what has that been your discovery, your observation?
One of the most amazing things I ever saw was a simulation of the universe that Harvard did. I think it was like 2014 on supercomputers, right? So it's like a data intensive computing thing like I do with, molecules. And I didn't really understand the scale. The dots I thought were stars in this big image. It was a movie of, that the computer generated to be a realistic view of galaxy formation in the universe. And when I realized there were galaxies, not stars, right? I was seeing the scale, and they still weren't zoomed out all the way, right? They zoomed out to like a big cube of the universe. And at that point, when you zoomed out, you saw these tendrils of dark matter that they can't see it, but they can infer it from gravitational pull. And it would look like neurons. They look just like neurons. So it's this connectivity pattern, this repeating patterns, fractal behavior that you see at the universe. I mean, the big time universe scale down, and you see it at the molecular scale too. We see it in our gene interaction and gene co-expression networks that we build for cancer and plants and things like that too. It's all there. It's just I think the universe is a repeating pattern of interacting things, right? And they're different. Like, there's a difference between a galaxy and a cell, of course, right? Practically. But the way things interact in this fractal-like behavior is you just see it if you zoom down to the molecular level or zoom up to the universe level. And that's where maybe that's what we're seeing, right? With we can't see the universe, we can't see the inside the cell, but with DNA sequencing technology and microscopes, we can see down and we can see up with the telescopes, and we have one of the most amazing telescopes in the world. And I, as a taxpayer, that's money well spent. I'm glad we're spending money on stuff like that to help us advance the species and see the way it is, right? It's beautiful. There's some things in life are absolutely beautiful, and the universe is one of them. That's for sure.
That's for sure. And I think today's guest, who we're going to bring on in just a moment, Josh, is going to be able to tell us about that synergy, that synchronicity that occurs both at the genomics cellular level in plants in this case, and at the, kind of more galaxy level in space, and some of the challenges of gravity and phototropism, et cetera. So with that, let's welcome today's guest, Dr. Josh Vandenbrink. Bring him on. Hello and welcome back. We have Dr. Josh Vandenbrink with us today. and we're really excited to talk about space plants. Josh, how you doing today?
I'm doing good. Thank you for having me on. I appreciate it.
Oh man, the joy is all ours, and you're coming at a really exciting time. Alex and I were just talking about the James Webb Space Telescope, but before we get to all that cool stuff, Alex, why don't you introduce our new guest?
Yeah, so Josh Vandenbrink, Dr. Vandenbrink, Dr. V. But we worked together years ago, and he's done many different things since we worked together, and he's a professor at Louisiana Tech right now, and amongst other things, and I guess a double father, recent father, I guess, and a not so recent father. But anyway, so Josh is yet another one of the people that I've met over the years. Just great scientist, great person, who doesn't really completely exist in the box. He thinks outside the box too, and he works inside the box. He can do both things, and so that's how I would introduce him.
Fantastic. And so, Josh, one thing I like about you, and I've seen a lot of the work you do in the Praxis platform and the virtual classes you create. I mean, you have this kind of very cool, modern, contemporary vibe about you, and you've almost branded yourself. Is it JVB? Is that the
VDB?
Cool... VDB.
Vandenbrink, yeah.
Yep, VDB. So you've got the VDB kind of. So from now on, you'll be known as Dr. VDB.
That's how I sign all my emails to my students. Yeah,
love it.
So that came out of two. It kind of had two parts of a genesis for that. So first of all, when I was in high school, my parents being so creative naming me Joshua, I had like 4 friends named Josh.
Yeah.
And so all my friends would call me Vandenbrink because of that. And so we just refer to all the Joshes by the last name. So, whether it's Smith, I'm not going to use their real names, but, so for the longest time I've been, all my friends call me VDB. And the other part is what's amazing. Well, I shouldn't say amazing, but for how phonetic my name is, like there's no like hidden letters or anything.
Right.
So many people get it incorrect that like, and I tell my students when I teach a class in person, I say like, what happens is they get to the van and they can do that, and then they get to the DE and they're fine there, but then their brain like freezes and they're like, is that an R or an N? And then is it Brink or Berg? And I get all these combinations. So I was like, just remember, you can. Yeah, exactly. Yes, yeah. I've been everything. So I was like, if you can remember those 3 letters after VDB, then we're good to go. So.
This is very similar to why I went the 4th. I mean, DJC the 4th, David James Clark the 4th. It was, there were lots of other Davids. I'm the 4th one. So all, my grandpa, my dad, they're all a bunch of Davids. And, you find out in school, you end up kind of branding yourself a little bit, and it sticks. And it's good. One thing we've noticed is it's good to stand out. So well done.
Go ahead.
No, I was just gonna ask you to tell us your story, but go ahead. You've already started.
Yeah. So, you're being a David and an Alex for that matter, a common name. So my wife and I, rebelled against that. And so my son's name is Arjen, which is, we got the name from a Dutch soccer player.
I like that.
Dutch. So we have Arjen, and then my daughter's Esme. However, Esme may now turn out to be a much more common than we thought, because every time we tell people our daughter's name's Esme, they're like, oh, from Twilight. We'd never seen Twilight, but I guess there's a character named Esme, so it's probably she's going to be in school with like 10 other Esmes now, which is, that's not our intention. But, yeah.
I don't know. Twilight was 10 years ago. I'm not sure it's as popular today, but yes, you're probably right.
Yeah, I hope not.
Yeah. So how did you get into bioscience, and what got you excited about space? And it's the biohackers love to hear about how people migrate into, more on the medical kind of cancer side versus the plant kind of agriculture side. We had, Dr. Markandeya talked about insect pheromones on the plant side, Dr. Alex Harkis, yourself. So fascinating, you start in bioscience, then you go space, then you go plants. Tell us how that all happened.
So when I was in probably, it must have been middle school, my grandma got diagnosed with Alzheimer's. And so I was interested in genetics at that point because I wanted to know if my mom would have it. I wanted to know if I would have it. And I was good at biology. And that was one of the few things that, I should sell myself short. That was the thing I was best at.
Yeah.
So I kind of hopped around and ended up at Western Michigan University for my undergrad. And at that point, I wanted to get some research experience. So I worked with the only lab that had space or a place available for me. And that was in with insects, genetics of insects. And so I was working with Monobia quadridens, which is a parasitic caterpillar hunting wasp. And so what we did was we looked, they have this really cool sex system. So Hymenoptera are like ants and bees and wasps and the insects of that, I guess, clade or family. And they have this cool sex system where fertilized eggs will always become female, and then male eggs, or unfertilized, will become male. And so it's called haplodiploid. So if you're diploid, you have 2 copies of your chromosome, you're automatically a female. And then if you're haploid, you're automatically a male. And it's the same with ants and stuff. So like all the workers are males that are unfertilized eggs, etc. And so what we were looking at was how these wasps, they have what's called a complementary sex determination system. And so they have an additional gene that kind of throws a wrench in this haplodiploid thing. And so we set out all these nests and these trap nests and stuff that would allow us to collect these insects, to collect the eggs, and then see how this sex system worked. Through that process, what I learned was that I don't want to work with anything that can hurt me. Because there was multiple stings. If we had, so what you do, you set these traps out, and then over winter, the wasp would lay their eggs in the fall. Then over winter, we'd go collect the nests, and then we would genotype the eggs and stuff. But if we got like a warm week or two, the wasps would hatch, and so we take them back to the lab and we open them up, and then all these wasps fly out. Catch them, because you can't. The big problem with it is you can't let them free and like get into other stuff, because you need to know the genetics of everything. So you have to like capture them or kill them, and then so you get stung. So that was my experience as an undergrad, and I decided no more. So I applied to a few graduate schools, and I got. I was down to 2 choices. One was to go to University of Memphis to work on Alzheimer's, which was my original dream. And then the other option I had was to go to Clemson University. And at the time, my now wife and I were dating, and she was looking for an undergraduate school, so we explored both options. And I was interested in enough things to say, well, I'll put the Alzheimer's stuff on hold, because I didn't, no offense to Memphis, but I didn't really want to live in Memphis. And Clemson is just a gorgeous campus. We're much more small town people, my wife and I, and didn't want to live in a city. So we ended up going to Clemson. There, I was originally interested in 2 labs, and those 2 labs were Dr. Hong Lo, who did transgenics in rice and turfgrass and stuff, and so I did a rotation with him. And then also Dr. Jim Morris, who was a parasite guy, so he works with Trypanosoma brucei, and so African sleeping sickness, so I did a rotation with him. And then I was like, man, I really like those 2 labs. Let's find a 3rd lab. And so there's this professor there. This is honest truth, John. Not just
if 2's good, 3's better.
Well, so we had a 3rd rotation. So there's this really cool professor there who I got along with great, and I really liked him personally, and I didn't know much about his research, but that was Dr. Alex Feltis here. And so I ended up working in his lab. in the summer, had zero intention. I don't even know if Alex knows this, but I had zero intention going into it. Like I wasn't closed off to the option to work with him, but I had figured I would choose those other two that I was really interested in. Went into his lab, absolutely loved it. Loved working with Alex and being his mentee, and so I decided to stick with him. We ended up we were doing biofuels research at the time. That was kind of before Alex has kind of pivoted to more the health stuff, but we were doing some like sorghum feedstock type biofuel, looking for fermentation genes and things like that. And so loved my experience there. I think we published like 8 papers or something like that while I was in the lab.
Yeah, you worked your tail off.
Yeah. So so learned a lot, and then graduated from there. I did my first postdoc at University of Virginia, where I've hopped from insects to biofuels. Then I went to evolution of sunflowers, and so we were looking at heliotropism, which is, sunflowers follow the sun across the sky, and then at night, without any sort of external cue, they know to grow back towards the east to wait for the sun the next morning. So we were looking at how this is possible. What's the components? And we ended up finding that there's circadian components to this growth cycle. And so really cool experience, kind of looking at the reason I got hired is because of doing these field studies like I did with Alex. And so we were able to grow all these different varieties of sunflower and things like that. So I was there for only a short time, 1.5 years. And my wife got admitted to a PhD program in Mississippi. And so Virginia to Mississippi, way too far. Can't drive that. Like on weekends. And so I started looking for jobs, and there was a job that came up for a space biologist. And that was the only job that was like at the university for a postdoc. And I thought to myself, who applies for this? Like, is there this pool of candidates of space biologists out there that are looking for this job? Or am I qualified? I had no idea. So I ended up applying, and I got a response from Dr. John Kiss, who was the project lead there. And he really liked that I was working with heliotropism because his space research was really focused on phototropism and gravitropism. So they're both plant-mediated movements. So a quick aside, plants can't run away or run to or move to get away from a stimulus. So tropism is a way that they grow in response. So if you've ever put a plant in the windowsill, you'll notice that it eventually, all the leaves and the flowers and stuff like grow towards the window because they want the light, right? So he enjoyed that or valued that I had experience with heliotropism, and I actually got hired, which was amazing. And so my wife and I could be in the same place. Yeah, it was great. So my wife and I could be in the same place, and I got to work on NASA stuff. So he was funded for this series of experiments called seedling growth. And so that was 3 different rocket launches that we sent seedlings or seeds to the space station that we germinated on board. And then because of SpaceX's capabilities to download, as they say, or return things from the space station, we were able to take back those seedlings after they had grown in space for, I think, 14 days roughly, and then do genomics on them and look at what genes are differentially expressed between Earth and things grown in space. And this is really, there's kind of 2 components. There's a theoretical component, is we wanted to look at how the interaction between light and gravity works in plants. So if a plant has, say, I'll make sure I can get my hand in the picture here. So if a plant has like a light source over here, it wants to grow towards the light. But gravity or gravitropism says that the plant should always grow its shoots away from gravity. So it's got 2 signals, one above and one to the side. How does it navigate that? Does it say go more towards the light go more towards gravity, find a happy medium in between. And so we wanted to find that theoretical aspect of how plants kind of respond to external stimuli. On top of that, NASA, they want to just know how to grow plants in space, right? Because we're looking
the Martian.
Exactly. So we're going to be sending people to Mars and the Moon in the next decade, supposedly. I guess like early 2030s. So they need to know how to feed people, because you can't keep sending rockets full of food to the Moon and Mars. It's just too expensive. And so if you can send seeds, use sunlight, use gases, we can get carbon dioxide release it from the rocks, or take it from the rocks of Mars and things like that, then it's a much cheaper way of doing it. So loved it. Still do space biology to this day. So I was there with Dr. Kiss for about 5 years. We got through all the seedling growth experiments, all the rocket launches. That's the operational side is concluded. We're not sending anything else up right now. But we still have a little bit of data we're working through. And then since then, I've moved to Louisiana Tech, where I'm still continuing on this space biology journey. I'm moving a little more to the computational side. I work with NASA GeneLab, which is they have this group called the Analysis Working Group, where it's just a bunch of experts from not only world-class organizations in the United States, but also abroad. There's there's members from University of Belfast and King's College and stuff in England, and some German universities. I think there's someone from Max Planck, I'm not sure, but all these really renowned researchers looking at all this genomics data that NASA's generated. And so that's kind of how I keep busy right now. But yeah, that's my story. That was a long-winded journey of what my story is, jumping around. The one thing I do want to say, if there are students watching this, and I always tell my advice to students, what you do research in undergrad or in grad school does not lock you down forever to what your career is going to be. There's so many students that are worried that, for instance, at Louisiana Tech, we have a fantastic researcher, Dr. Jamie Newman, who does stem cell research. And they're all interested in medical genetics, genomics, medical research. And they all think they have to get into that in order to do that as a career choice. But that's not the case. You can arm yourself with the skills that make you a productive researcher and understanding how to ask questions and address questions, and you can jump anywhere. Like Alex can add to that. I think you started in cancer research, right? And then you went to biofuels and...
Well, yeah, and to go on that, so like I started off as a wet bench scientist. When I do my PhD, that's what I wanted to do. I wanted to cure cancer, and I pipetted liquids into tubes and saw tubes that were clear liquid and was frustrated if there wasn't anything in the tube. You can't see it. You have to go in there and look at it. It's just like, it's really, really interesting. But I shifted over to computational stuff because I love genomics when the human genome came about, and I started doing a lot of the computational work. And so it's been, you're the last person in my lab that was like doing really heavy field work and wet bench work, and we're almost completely computational now. And so what do you think about that? Like, I see this, for me, it's been like, all my time goes into that. I don't have time to do the wet bench work just for practical reasons. But there's so many discoveries and experiments that can be done using data from, like, maybe for example, like using GeneLab, for example. That's all open data, right? That anybody in the world can download that, and it's things that were in space, right? That's mice and rice, right, and everything in between.
Yeah, it's a fantastic resource. So starting with your second question first. So like GeneLab, last I checked, I was on there a couple weeks ago messing around with some data. I think there's like 400 different data sets from different organisms. There's a lot of mice stuff, Arabidopsis stuff. The thing you have to remember is because it's spaceflight stuff, generally it's small model organisms, right? So there's no corn or anything because there's no room to grow that on the space station. So it's mice and rats and Arabidopsis. So there's a lot of stuff there. For your first question, the the marrying or the finding the balance between computational biology and wet bench biology. It's hard. It's hard to navigate because part of me, I love doing the computational stuff, but there's so many times I found, especially in space biology, or I should say in plant biology, going to these meetings where people do a bioinformatics experiment and they identify genes and they say, this gene must cause this phenotype to occur. So this gene knocks out your gravity response, just staying on brand here. And then that's it. And then they never follow it up, and they never, it just dies in the literature, or people take it as canon or whatnot. And so I think it's really important that when you identify these genes to actually do the experiments to confirm that you have identified something that's actually truthful. And so what we kind of do, what I've kind of whittled down my wet lab to, because COVID's really like put a damper on like the momentum of my lab.
Right.
Having everything shut down. So what we're doing really is we've done these RNA-seq experiments, and we're going to just knock out these genes that we identified as differentially expressed in space and the ground, and actually really grow the plants and see if we can see the change that we predicted with the RNA-seq experiment. Because a lot of these bioinformatics experiments are almost like hypotheses for wet lab experiments. So you've concluded and you've gotten a result that says that this is the case, but is it really the case? Like, all right, now let's see if we can actually do it in the plant as opposed to in the computer. So that's kind of the balance that we're trying to strike in my lab.
What do you think about? So, I totally agree with you, and you have to verify things in some way, right? Mm-hmm. Anything in science, especially in biology. There's not a lot of experiments that have been done in space because it's expensive, and there are some other experiments I guess you can validate some of your experiments with. But there are, like in the National Center for Biotechnology Information, NCBI, this is the U.S. database of a lot of genomics data, and there's counterparts around the world that now there's like, I haven't checked it in a year or so, but there are 4 or 5 million experiments. I wouldn't be surprised if there weren't 10 million now. And so what we've started to do is we go through, we do our hypothesis generation. We find some cool genes associated with traits, basically what we do in the lab. And we're a little bit kingdom agnostic, right? We're doing plants and animals too in the lab. But so if you go and verify on the computer, you find something using a computer a data set you download from the public sources, and then find a pattern, a gene or something, and then go verify with another gene set. Like, and I'm not that's a approach we're taking. Does that seem reasonable to you as far as being like real experimental biology? Because a lot of people don't think so. Because once it's done on the computer, it's not real.
Yeah. I've got those headwinds too from people.
Yeah.
Now I understand, and I didn't want to when I said the stuff about people stopping before they finish the experiments, so to speak. That's very specific to the plant stuff that I'm doing because it's in Arabidopsis. There's so many resources for Arabidopsis. So Ohio State has this Arabidopsis biological research network where they have a seed bank where they've knocked out just about every gene in the genome. And you just have to order it and grow it and phenotype. So it's not like heavy legwork to confirm these things. But if you're somebody that works with human stuff, like Alex, you do a lot of cancer and stuff like that, you can't go knock out some person's genes and then see if they get cancer or if they're cured from cancer or whatever. So the avenues running training and testing data sets, kind of like you're talking about, where you take a large sample, or say you take 80% of the publicly available data sets, you build your model, and then you test on 20% to see if you confirmed what you predicted. That's completely that's great. That's because you can't, and same thing with space, there's just not that much. I can't, if I come up with an experiment that is super space specific, that I can't, like, I have Praxis for gravity stuff I can do in my lab.
Yeah.
But I can't just be like, oh, I found these genes. Let's quick throw it up to the space station. Like, it doesn't work that way, you know? It's like
now,
a 10-year
maybe
process.
soon.
Yeah, maybe. Yeah, I just gotta, win the lottery of, Jeff Bezos putting these celebrities. Maybe I have to become a big enough YouTube star to be on one of these rockets, like Michael Strahan and whoever else, William Shatner, and then I can just take my plants with me.
JVB?
Yeah.
That's a great point. I mean, when we were chatting about the show before, I was like, hey, you going to visit your plants up in the space station? I mean, is that a bucket list for you, or would you love to go up there?
Yes. It's been beaten out of me by my wife. Like that. I had toyed with applying, but now that I have kids, that's kind of changed too. Your perspective changes a little bit. Like maybe, if I'm not too old when they are adults, I got started late with kids, so I'll probably be too old, but I would love to. Like if my priorities were not children first, I would be signed up and trying to get my butt up there. But it is, regardless of how much technology is advanced, it's still pretty dangerous. Like I remember I watched the Challenger documentary that was on Netflix, and I think it was like the odds of an astronaut dying back around the Challenger time was like 1 in 60 or something. It was like crazy high because they had the problems with it might have been right after Challenger, because that took into account the 6 people that died in Challenger and then the people that died on the launch pad during the Apollo program and things like that. Right. And there's still, SpaceX has been very successful with not having any sort of malfunctions with their crew program. But when I was doing research, there was a flight to the space station that was, I believe it was CRS-10 or something like that, which was a cargo resupply mission to the space station. And we were scheduled for number 11. And that was the one that blew up on the way to the space station, which was probably 5 or 6 years ago, maybe even longer than that. And I remember texting somebody and saying, oh, thank God that wasn't my flight, because this is like years in the making of doing these, getting these experiments. Yeah. It's like, it doesn't affect me. And then I got an email from the European Space Agency, some people we were working with there, and they were like, bad news, some of our equipment was on that rocket. And so we lost, there was a bit of a delay because my seeds weren't on there, but stuff that was vital to the experiment. So it also kind of put into perspective that spaceflight is still pretty dangerous. And SpaceX has had a few go up, not recently, but I think they're getting better at it. But
see, I want to ask about that. So you remember you telling me one time that what is so this is now, and then I'd like to talk about in the future with space experiments, that what was the lag time from writing a grant to getting something on the International Space Station, which is a reality, and what was the cost? Was it their cost per kilogram or cost per gram, the base cost just to get the stuff up there?
So I think before we even got a rocket launch to the space station, so first of all, NASA's turnaround time on reviewing grants is pretty long. So for instance, grant deadlines are usually like February, then you don't hear back until October or something like that. But I think between the acceptance and the first rocket launch may have been 3 years.
Wow. Is that pretty standard, or was there, because there are any events that happened?
That was before there was even any rocket issues. And so, because I think the first experiment that went up to the space station was on commercial resupply 2. So it was it was either 2 or 3, I think, really early in the commercial resupply. So they hadn't had any blow up yet. and then beyond that, everything with the space station comes down to crew time.
Yeah.
So there, like,
Gotcha.
the upload and the download, and things like that, that is a concern, whether it is, how much space you're taking up, or how much things weigh and things like that. But the real bottleneck is how much time the astronauts have to do your experiment. And so that is a big holdup. And so sometimes you can go and do a flight where you fly up to the space station, and then they kind of put your experiment on ice, and they're like, this will be done when somebody miraculously has time. And so I know Amara Pereira at NC State University, she went up on an experiment like that where she had to wait. And luckily, one of the astronauts, I wish I knew which one it was so I could give him credit, they have scheduled free time, and he or she was willing to do the experiments in their free time to help out some of the researchers.
Oh, wow, that's cool.
Yeah. But that's the big thing with getting funded too, is that NASA, everything it's super military. Like, NASA was born out of the Air Force, and so it's like very regimented, just like the military, in that, like, the astronauts, almost to the minute, their schedules are, documented for like the entire time that they're on the space station. And so when they send out these solicitations for proposals to write, to do your experiment up there, they know exactly how many they can fund, and they also know how much available crew time, or how much crew time is available, and they have to marry that with which ones they're going to fund and things like that. So it's amazing the amount of red tape that the telecoms or conference calls that I was on during these during this NASA work, I can't even count how many. And we were doing a joint experiment with Dr. Javier Medina, who is a researcher in Spain, who recently is retiring, I just found out, which is a loss for the space science community because he's fantastic. So we had to also do the logistics between NASA and the European Space Agencies. You have these two governmental agencies that are butting heads on things and arguing who has responsibilities for this and that. It leads to delays, and it leads to just tremendous amount of effort for what it's fantastic when it's done, because it's exciting to see a rocket launch and stuff, but it's not easy peasy. Like, if you are somebody that's impatient, don't go into space biology. That's my answer.
Well, good advice.
But that's the future. Do you think it's changing? I mean, so like if SpaceX, for example, they're creating lots of, they're mass producing starships potentially. I guess the new Starship is up for an orbital test pretty soon, right? I don't know if they got cleared for, I haven't looked at it in a month or two. Did they get cleared to do it?
I'm not certain when their orbital tests, their dates have changed a bit.
Yeah.
Like, the pandemic really hit them too, and threw off kind of everybody's schedules. So I know that they had their belly flop landing. That was a while, within the last year. But I'm not sure.
But they're supposed to have a full multi-Raptor engine test, right? Of the a real deal kind of thing. Let's say they pull it off. Let's say 5 years from now that they're able to, at least send one to the moon robotically, or even to Mars, right? Which doesn't seem too far out. And so if the floodgates open, right, where there's lots of people in space, probably with time on their hands, right, if they're traveling to and from other planets, there could be a golden age for space biology, right? Pretty soon, because it seems like it's going to explode. Well, I guess I shouldn't say that. It's going to it's going to take off.
It's going to accelerate in progress.
Accelerate.
You'd hope so. Something needs to happen like that, though, because NASA, the space station's only going to last in another 10 years, and you're going to crash it into the ocean, and it's done for. Well, there's been talk since I started doing space stuff of them finding a commercial partner to take over the space station. So we'll see how that works.
Yeah, but also, that's a bunch of tubes, right? Like it's not a industrial complex, right? Like
it's funny because it's piecemeal together. So first of all, you have multiple chefs in the kitchen, so to speak. So you have like JAXA, the Japanese Space Agency built a module, and then ESA has a module, and NASA has a module, and then Russia has like half the space station, which in this political climate has been a big nightmare. And previously, with the when I was working on the space stuff, there were some issues with Russia, and I think it was like the Syria stuff, where NASA and ESA said, we're going to continue to work through this, but whoever's in charge of Roscosmos now, which is the Russian Space Agency, he's much more politically motivated than whoever was in charge at the time of the previous tensions. But so this is coming to an end, this space station era. So there's talk of there's a couple different projects. There's another project called Gateway, which is I believe they're trying to build a space station that's not in low Earth orbit, or LEO as they say, like around the moon or something, which would be the gateway for them to go to Mars. And then there's also talk of a moon base. And the issue though becomes that NASA's directives change with every administration. Yeah. And so through the 90s, luckily, when Bill Clinton was in office and his administration, they finished the space station. Then when George W. Bush took over, he wanted to go to Mars. And then, so they scrapped a bunch of stuff that they had. And then when Barack Obama was in office, he wanted to go to near-Earth asteroids. And so then the Mars thing got put on the back burner. And so those programs, their funding gets yanked. And then they did the near-Earth asteroid thing. And then Trump comes in, and then he wants to go back to Mars. And so then they're like, well, we just spent 8 years doing this asteroid thing, and now we have to go back to our Mars plans. And so then, Biden, now it seems like Mars is still on the table, but the moon is more attractive because Russia and China want to, they've been really pushing to go back to the moon. And so now we're like, okay, well, maybe we go back to the moon. And so it really, it makes it a difficult landscape to navigate with what NASA is doing and how your research can help them. And so when their focus changes, their funding changes. So when you go to near-Earth asteroids, they're not really worried about growing plants. If you go to the moon, you're more worried about growing plants. And so, so hopefully this private space thing, getting all the way back to the private space that you asked about, hopefully that opens that up a little bit. And, I'd like to see SpaceX or, Blue Origin and stuff aren't really into the deep space game yet. But I'd like to see them address some of these biology questions. So SpaceX and Elon Musk seem very intent to get to Mars, but I haven't seen anything biologically focused from them. It's all engineering stuff.
Wow, I wonder if they're going to the granting system mechanisms shift from the government to a company like that. It makes a lot of sense. I mean,
but what's going to be interesting though, they're going to want it as patented proprietary information. They're going to if they're funding it. I mean, SpaceX or Elon Musk's been pretty open with this. I know, like when his Tesla, they released the battery patents, so maybe he's a little more open to sharing ideas. And it's not like he has a ton of competitors, that are going to scoop his technology. But it's a question. I am a fan of what Elon and SpaceX are doing. This stuff, their technology is amazing. It was awe-inspiring. Watching the rocket launch for our seedling growth stuff was very cool, and the power of these engines and stuff. But the thing that blew my mind is watching the booster land afterwards.
Yeah.
So we got to,
It's magic.
we were and our last rocket launch was the first reused booster for SpaceX, and so a refurbished one. And it landed, and you could actually see it from the VIP area that we were in, which was pretty cool. Not like I'm touting that I'm like this big VIP.
You're a space VIP, Josh, and you're on our show. This is so exciting.
Yeah. You know what also is pretty cool? At that same launch, Buzz Aldrin was there. And he gave a talk to our little group of the scientists that were involved with the rocket launch. But man, NASA, like, I've never seen. It's like he has his own secret service. So he came up and he gave a little talk, and then before any of us peasant scientists could go talk to him, they like whisked him away to some like secret location. So I got to hear him speak and be in the same room with him, but then he was out of there. But I mean, it's been a cool experience. I've gotten to meet a lot of astronauts. Charlie Bolden, who was the head of NASA for Barack Obama, who was a former astronaut, he would be at the meetings, and Bob Cabana, who was the head of Kennedy Space Center, also a former astronaut. Super friendly people. The one thing I've told a couple of my students who are interested in NASA jobs, it's very intimidating. Like, NASA is intimidating. You see like Big Bang Theory, Jet Propulsion Laboratory, and think all these brainiacs that are like super, cold and mathematics and etc.
Yeah.
Everyone I've dealt with at NASA has been the nicest, most helpful group of people. And I don't know if it's because they all are like living their dream, and it's just like, I know there's like a lot of red tape and stuff you have to go through, but it's still, you're working at like the coolest organization on the planet in essence. It's like, it's awesome. And they've been nothing but nice, even all the way from Charlie Bolden and Bob Cabana and the guys that run everything has been nothing but nice when I talk to them, to the people that run Ames in California. Everyone's just, so if you're interested in doing NASA stuff and you're like, where do I start? Reach out to somebody at NASA that like does what you're looking at, and odds are they'll reply and be very willing to talk and nice.
Would you go, like, if you were, let's say you were, looking for a job, you're getting your degree, undergraduate degree or something like that, and you wanted to go down that path, would you look at SpaceX? Would you look at the private sector and NASA? Or, like,
it depends.
Is it mature enough for that?
It depends what you're doing. I actually just went through this. So I have a student who is just graduated with her bachelor's.
Yeah.
And she was looking for any avenue to get into NASA. And so I'm very, I have connections in the biology side. I don't have a lot of engineering, like the James Webb.
Yeah.
I've never met anyone that works with James Webb. I think it's cool and I follow their stuff. And I've been to JPL and I got to like meet a couple people at JPL, but I don't know any of the rover team or anything like that. So I'm like really, sectioned off. Like NASA's, there's like 50,000 employees, so it's a big organization.
Oh wow, I didn't know it was that big.
Siloed.
So we, so the student was looking for avenues to get in, and going back to NASA being kind of an offshoot of military culture and stuff, they have contractors, kind of like military contractors, but contractors that take these contracts to do their biology work. And so previously it had been, I think Lockheed Martin, and now it's KBR, and these different entities that take on these contracts. So people can apply to work for KBR at NASA and then jump from there to the NASA side because they're kind of like quasi-employees at that point. So Caitlin was looking, my student Caitlin was looking at how to kind of get in that way. And so there's avenues that way. She ended up actually just getting an internship at Kennedy Space Center. I'm super proud of her. This is like 3 days ago that she texted me and said, and she,
congratulations, Caitlin.
She told me, Caitlin, wherever you are, I'll have to sign this direction. We talked about you on the podcast. But one of the things is that the person that she has the internship with liked that she was doing the Arabidopsis stuff, not necessarily space biology stuff, but also working with Arabidopsis. But if you are somebody that is more into the engineering stuff, definitely go for SpaceX and Blue Origin and Virgin Galactic and Orbital Space Sciences. I think they just changed the name, Orbital ATK or something like that. All these companies, and there's also even smaller like ancillary companies. So like one of the companies that we had a lot of interaction with is this company called TechShot, and I believe they're based in Madison, Wisconsin. But while they don't make rockets or anything, they make components that go onto the space station. So they make the machines that do the work on the space station. So there's a ton of opportunities in engineering. Biology is a little more narrowed down, and a lot of that is NASA does a decent amount of biology research at NASA centers. So Ames in California, in the Bay Area, and Kennedy are the two kind of biology research hubs. But a lot of their research is actually farmed out to universities and through the grant system. So if you are interested in getting a job at NASA, it's get your foot in the door. That's my biggest, find an internship, work at KBR, do the summer. They have a ton of summer internships or summer programs from high schoolers all the way up to graduate students. Get to know some of the people and sneak in, and don't be intimidated by them. They're super nice people. I'm gonna say that, and then someone's gonna watch this and be like, I knew the biggest jerk NASA person.
Well, to spin this a little bit towards this biohacker thing, I want to ask you something that we're doing. And we're definitely focused in biosciences, and motivations like doing space biology. That's like a fantastic place to go, even with its limitations on the number of experiments you can do and all that kind of stuff. But one of the things that we're really interested in doing, and I'm interested in this personally, is just training people how to code and not be a computer scientist necessarily, right? But just know how to do some coding and be able to use advanced computers. And some of these are democratized systems that people have access to in the U.S. as U.S. citizens can get access to them, and be learning how to do data mining in large data sets. And a lot of that involves machine learning, artificial intelligence, and stuff like that. And it seems like it's not a very big stretch you could do bio, if that's what you love, and learn these skills. And then they can be applied to other things too, right? Once you know data science, AI, I mean, you'd have to go through engineering programs, right, to be an engineer, but there's probably other aspects, I know there are in the financial sector, the accounting division of these places, right? So there's other ways to get into the system if you know this compute. And I've always been telling my kids and other people, major in what you love and learn Python, right? Because then you have the ability to get a skill with that. So what do you? So I know you're teaching a lot of bioinformatics and using the R programming language to do that. And so how do you feel about that philosophy? I mean, I'm going to go be as extreme as I can. I think everybody in the 5th grade, you shouldn't be able to leave middle school unless you know some Python.
Man, that might be extreme.
Yeah,
I eventually
that might be ridiculous. So.
Well, no, but I mean, and it could just be because I'm old, and I remember taking typing class, and I'm sure they don't do that anymore because kids already know how to type since they're born. It's born. Yeah, being in front of a computer. No, I think it's a invaluable tool. I really do, and I've preach it to my students. I tell them, whether you are going to be a doctor or a researcher or whatever else, this can be useful to you. And I'm teaching a class this fall on Praxis called Data Products, and kind of the mantra or the essence or theme of the class is that, all right, you've done most of my students are pre-med, so probably 95% of them, and so they're just getting into this brave new world of research and bioinformatics and computational biology. So I phrase the question, okay, you've done a clinical study and you have all this raw data. Now what?
Mm-hmm.
Yeah.
Where do you go from here? And that's where data products kind of takes off, is that, okay, we can use, in this case, we're using R, and you can take that and you can do preliminary data analysis, you can describe your data, you can graph your data, you can do all these different things to kind of get a hold of what the story your data is telling.
Yeah.
And if you're a doctor, that's just as important as if you're a researcher. And I think what's been one of the bright sides, if I hesitate to use the word bright sides, but one of the bright sides of the pandemic is that the amount of information, that public health information that was conveyed to the general public was just outstanding. Like New York Times and all these different databases would show like daily updates and all these different things. Yeah. And I asked the question to my students, what is the point of doing research if you don't convey it to anybody? Like, especially if you're doing health stuff. You've got to get that out to the layperson. And so these tools allow you to do that. And so whether you're a doctor or a financial person or whatever, Python and R are really built for you and can be applied to almost any, career path.
Yes.
It's just a valuable skill.
Yeah, and I think throwing in there like having a large data set that you're trying to find patterns. It's something things that don't fit on your laptop typically. Mm-hmm. And so you need a computer to be able to process it, right? And a lot of that's in the cloud. And so that's where I'm really trying to be sort of a prepper for these things, right? And being able to get people to learn how to use the computer, the system, and then, be motivated by a cool problem like space biology, right? I mean, it's just such a cool thing and very, very useful if you're trying to eat when you're on Mars, right? Like it's it's really interesting that are we really going to, there's no beam. We can't beam up the food. They're going to have to grow it. So it takes development cycle. But anyway, just a motivation mixed together with all this stuff. And then the skills are translatable to different fields, right? It doesn't have to just be bio. So that's where we're trying to do in these hackathons and we want, I want to get your students to be participating in the fall. One's really into it. It doesn't have to be everybody, just the go-getters.
Yeah. I'll have to advertise your biohacker stuff. I just got through my first class, so I'm still like, kind of first course. But I'd like to start pushing them into your biohacker thing. The one thing too with it adding on is that there's stuff you can do that's even fun or if you want, it's kind of nerdy to have it like as a hobby, but like one thing we're going to do in the class is text mining. So like we're going to go on to Twitter and download all these texts on a certain day and then make word clouds and you can measure sentiments and that's become a really big thing in politics. So measuring sentiment of voters and things like that. It's a big thing, and for this pandemic thing with anti-vax versus vaccination and all that stuff. So social media and all this stuff, R and Python can provide avenues into a lot of different fields, and even ones you wouldn't even think, politics. If you want to get into that.
I would love it though if that was watching the sentiment is a great way to see how well your propaganda is working. Right? Just one application.
Right.
But the other way, if you're into the data, is I find out if it's true or not, right? And just trying to I think we really need some kind of tools. I like to think of the tools in the natural world to find out the truth, underlying biological systems, right? What are those genes that are doing it? But I think in society too, just a little bit of this instead of I think it's real or not, it's fake news or it's not, I believe this theory or not, just a little bit of a p-value, some idea that this is actually a real, we need this, right, to know what the truth is.
Yeah, it's going to be interesting. So since this is their second class, I'm going to have them kind of pick their own adventure of what sentiment or whatever they want to analyze. But it's as somebody that's into stats, I see so many flaws with it already. It'll be a fun exercise, but I mean, you're only measuring the loudest voices on both sides, right? You know, so you have this bimodal distribution of sentiments.
Exactly.
Like the large majority of people in the middle don't care, but you have the people that are like hate vaccines, or the people that are like want vaccines, or Democrat or Republican or whatever it may be for any ideology. So, you know, you're not capturing the full thing, but it's still going to be a fun exercise.
You're going to get some skills. I guess I would a little bit dark with it, but it's also like...
I wish I knew a little bit more of social media. I'm just dipping my toe. Like, I've done a little bit of it. I wish I knew a little bit more of like how these algorithms work to figure out what's true and what's not true. And confidence intervals.
Well, that's interesting because like there's a lot of the tools that we're using that I understand the input and output pretty well as a you know, a professional scientist. I've looked at this stuff for a long time, and I can feel my way through it and then prove it with experimentation. Right. But there's a lot of black boxes in there, and that's where I have to have interdisciplinary people on my team, right, to be able to do anything. And I'm not ever going to understand everything, but I can't. You can't be a jack of all trades alone, and you won't get anything done. But you have to kind of like have the knowledge of a jack of all trades to communicate with everybody.
Yeah.
You know? It's a hard balance to meet.
Yeah, that's there's kind of I wouldn't say age old, because it's not like bioinformatics has been around for, you know, hundreds of years, but there's, you know, there's this issue that a lot of scientists have had, and it's been relayed to me a lot, that they'll be doing biology and they'll reach out to a computer scientist or somebody to run their RNA-seq analysis or microarray or whatever they were doing, and then get the results and not understand the results, and then the computer scientist person doesn't know how to convey them or didn't run it right because they didn't understand the biology, and there's just too much of a divide. And so you have to be, like you said, you have to be not be able to do all the work, but understand the work to be able to communicate. So like that's kind of where bioinformatics, like understanding, even taking a little bit of bioinformatics class. So if you do lab research in the future and find somebody that does your computation for you, you understand enough to ask him questions or ask him questions and things like that to be able to communicate, because there can be too much of, you need a little bit of that, you know, trivial knowledge to make sure things are collaborative correctly. Yeah.
I think we have a lot of medical students too, and that I go through our programs, and I make that argument with them of like, this may not be something you'd learn more deeply in medical school, like learning Python and stuff like that, but they are being faced right now with genomic medicine, and you need to be able to communicate with, you know, the bioinformaticians, right? They're already on the teams of a lot of hospitals and stuff, and just being able to have a conversation with them. And also the scientists too, because I mean, doctors are artists, and they know how to, you know, cut into somebody and take stuff out and put stuff back in. And I would be there, you know, crying in fetal position, crying to even think about how it's started, right? But they get this stuff, but then so they also need to talk to the scientists too, right? I think there's a technology scientist, you know, clinician, like partnership, that if that becomes standard. And I would throw in, if like the data was a little bit easier to obtain for the scientists, it's very difficult to obtain clinical data, right? You have to be an elite institution. I think it should be open a little bit more to everybody personally. But like this interdisciplinary stuff, I think will lead to, you know, sick kids getting out of bed, finding cures for them and stuff like that.
Yeah.
That's a big motivation for what we're trying to do.
So as we think about the Praxis Pyramid, and, you know, one thing we've kind of alluded to, Josh, is this concept of your students, you know, the base of the pyramid is training, and that's, you know, where you're a real instrumental part of what we're doing, and Alex, of course, as well, and trying to get people these skills, the Python and the R and understanding. And then the next, you know, step up the pyramid is research, you know, getting them into biohackathons and scaling the research in a virtual way by, we have these Discord servers that are dedicated to doing particular experiments, and people collaborating in the Praxis platform and Discord and other places. And then ultimately the pinnacle of the pyramid is workforce enablement, getting them these great jobs, as both of you have done. You care enough about your students that you help the Caitlins get those really good jobs at NASA or wherever it is. And so being able to scale that virtually and make it available to underrepresented groups and people who may not typically have the funds to go to an elite university, et cetera. So that's really the BioHacker mission. So as we wind up the podcast, and thank you so much, this has been a fascinating conversation. Coming back to that, what is a biohacker to you? Because you're an instrumental part of that pyramid, and you are really at the ground floor, helping train the biohackers and then move them from research to jobs.
Oh man, I didn't know I'd have some tough questions at the end here.
Just one.
Yeah. I think really my mission, my goal is to get students exposed and not terrified of bioinformatics. That's my first step. Because so a quick story, because I know we're trying to wrap up, but when I interviewed at Louisiana Tech, I met with a group of some undergraduates, and they asked you the interview questions, and one of the interview questions that they asked me was, what class would you offer that is not currently offered at Louisiana Tech? And I said, I'd love to teach a bioinformatics class. And the student said, oh, that's cool, I wouldn't take it. And so and I said, why not? And she said, it just sounds too intimidating. And so it's been my mission with the way I design my classes and the way I present my classes to take that intimidation away from bioinformatics. Because to make a biohacker, most of these students, the motivation is there, right? They want to solve these problems. They want to address these issues. But I think a barrier to entry is the intimidation of coding. Like, it's coding scary, all this stuff. And so I think for me to create biohacker, or for my students to become biohackers, is first and foremost to take away that intimidation and to show them that they can do it. These are pre-med students. They're not in any way incapable. I mean, they're all really smart students. They're going to go to med school. It's just taking away the intimidation of doing that. And then once they get away from, being scared and actually getting into it and seeing the utility and having fun and doing things that, we play with real data, so getting real, results. Right. I've had students tell me at least, they haven't, graduated yet, but tell me that they're thinking about going into computational biology now because they think that they can do more good by being a computational biologist, and they realize they have kind of a skill for it to address these issues. And so, so I think creating computational biologists, providing access, like as you guys are trying to do, providing access and taking away the barriers, and one of those barriers being intimidation, to actually just go and do it, because it's not terribly hard. Anyone, put your mind to it. Anyone can do it. It's not, rocket science to tie it all back in, right?
Yeah, well done.
People they love it when they start doing it. I found almost everybody, even the people like, I can't stand coding. I'm going to medical school. I don't want to take this class that's required. They just can't stand it. By the end of the semester, they love it. I mean, maybe I'm delusional, maybe they're lying to me, but I really think that they get transformed. And I call myself a computational biologist to describe myself, but I'm a biologist. I care about biology, and those are just tools, molecular biology. I've done all that stuff too to get the job done, right? And so I'm looking forward to a future where we don't have to say computational biologist, because it's all built in, right, into what you're doing. You can't really get away, especially with genomics now, without knowing bioinformatics and computational biology, right? So, thank you for doing it. Let's keep the movement going.
You know what? I gotta say, your class is fun. I mean, you've got kind of that gamer thing going. You walk them through the labs like you're on Twitch, and you know, and it feels like they're playing a video game, but they're learning our studio.
The lighting going on. The
lighting is very cool. You can see in the background. You know, you've got that YouTube star kind of presence about you, and the cool VDB logo everywhere. I mean, if you're interested in having a fun contemporary experience and learning bioinformatics in a non-intimidating way, check out prxaistore.com and purchase Josh's course. And that's the first layer of the pyramid. And then, once you have some skills, check out hackers.bio and jump into one of Alex's biohackathons. I think you have one next week, Alex. There's a couple more coming up.
They're regular. Yeah.
They happen every month, and they're free. And you get an opportunity to do real research, publish papers, meet other biohackers. So it's an exciting community. It's a really exciting time. Thank you so much, Josh, for joining today. We learned a lot about NASA and space and plants and the bug sexism. I mean, all sorts of really cool stuff today. And with that, I'll bring it to the closing song. Take it away.